Death follows a highly organized, cascading biological timeline that begins the precise moment the human heart stops beating. This textbook from Harrison's Manual of Medicine outlines the foundational clinical benchmarks for this physical breakdown. The sequence begins at T0, cardiovascular collapse. This is the moment the heart muscles ceases to pump and systemic blood circulation stops. Without flowing blood, oxygen delivery to all tissues halts immediately. Physicians define this state as clinical death, a window that lasts for a brief 4 to 6 minutes. During this critical window, cardiopulmonary resuscitation can artificially force blood through the system, potentially reversing the dying process. When this 6-minute window expires without intervention, the brain suffers permanent, irreversible cerebral damage. The clinical pronouncement of death triggers an unstoppable biological countdown. In the seconds following the expiration of the 6-minute window, consciousness is lost as the cerebral cortex shuts down. Medical criteria for brain death require evidence of widespread cortical destruction and the absence of global brainstem reflexes. This diagnosis relies on proof of complete apnea, signaling the destruction of the medulla's respiratory drive. This combination of absent oxygen and absent blood flow is defined as hypoxia-ischemia. From the brain, hypoxia-ischemia spreads through the body, starving the lungs, liver, and kidneys. These organs fail on a staggered schedule, based on their individual oxygen requirements. While brain tissue expires within minutes, the liver and kidneys endure significantly longer. Biological death is a fragmented process, where systems fail at different rates rather than all at once. Inside individual cells, the crisis begins with the structure rapidly destabilizing and swelling. Deprived of oxygen, the cell switches to anaerobic metabolism to generate energy. This survival attempt causes a rapid buildup of toxic lactic acid that poisons the cell. The cell eventually burns through its last remaining units of stored energy, reaching a state of ATP depletion. ATP depletion causes the mechanical ion pumps in the cell membrane to fail, allowing surrounding fluids to rush in. This influx causes the cell to swell until the membrane ruptures, releasing internal digestive enzymes into the surrounding space. This process of cellular self-digestion is known as autolysis. At the microscopic level, the body actively dismantles itself from the inside out using its own biological tools. Over several hours, the microscopic chaos of autolysis produces visible physical changes across the body. Pallor mortis marks the loss of capillary circulation, followed by algor mortis as the body cools to the temperature of its environment. Gravity pulls blood into the lowest tissues, causing livor mortis, while ATP depletion causes the muscle fibers to lock in the stiffening known as rigor mortis. During the stage of putrefaction, hungry internal bacteria escape the confines of the intestines and rapidly begin consuming the body's remaining soft tissues. This predictable cascade allows modern medicine to temporarily intercept the process and save the living. Organ donation protocols use specific criteria, like donation after cardiac death, to identify viable transplant candidates. Transplant surgeons rely on the staggered timeline of organ death to harvest kidneys and livers before autolysis can destroy them. While this exploration is strictly biological and forensic, the clockwork biology of death is exactly what allows us to extract new life from it.